It's my great pleasure to introduce tonight's guest speaker, marine ecologist for Channel Islands National Park, Stephen Whitaker. Stephen is, as I said, a marine ecologist for the U.S. National Park Service at Channel Islands. He has been studying coastal ecosystems in Southern California for over 20 years. In his current position, Stephen is responsible for monitoring the shoreline habitats at the islands, including Rocky Intertidal reefs and sand beaches. Stephen also logs dozens of dives annually, monitoring the kelp forests at Channel Islands. For his master's thesis at California State University, Fullerton, he investigated factors affecting restoration success for the intertidal rockweed alga, Silvetia compressa. Hopefully I said that right, Stephen. In 2018, Stephen returned to graduate school to pursue a doctoral degree at University of California, Santa Barbara, investigating the patterns and long-term trends in the abundance and distribution of foundational species on rocky shores. Concurrently, while working and schooling, he is collaborating with colleagues across multiple institutions on a large-scale project to restore rockweeds across California. Stephen is a busy man, y'all. Other crucial things to know about Stephen is that his favorite pastime is playing his mandolin, very important to know this, that he is 2 months from the pound, and that he loves and has worked with horses. So without further ado, I give you Stephen Whitaker. Take it away, Stephen.
Excellent. Thank you so much for the introduction, Sam. Let me share the screen real quick so I can get you guys started on the presentation. And let me know when it shows up for you.
I see it, Stephen.
You see it? Okay, excellent. All right. Well, first of all, good evening, everybody. I'm just, first of all, blown away that you're taking precious time out of your schedules to be here with me tonight. And I'm actually pretty ecstatic. You may not be able to tell, I get nervous pretty easily, but I am ecstatic to be here tonight to talk to you about what may be the most dynamic ecosystem on the planet, a place that I've spent many years studying, the Rocky Intertidal Zone. And some people call it tide pools. So just one glance at a colorful tide pool, and you can easily see that these ecosystems harbor high diversity of seaweeds and animals. Yet these areas are constantly changing. And tonight, I hope to provide a brief glimpse into some of those changes using the most recent and advanced science. But before I can do that, it's first necessary to acknowledge that I'm representing a ton of bright folks tonight. Pictured here are some of my colleagues from the partnership called Marine, or the multi-agency Rocky Intertidal Network. I'll provide a more formal description of this group of intertidal researchers in the coming slides. But I wanted to take this opportunity to show my sincere appreciation for being part of this group of top-notch scientists. And they're just simply good human beings all together. So much of the data that I'll be presenting tonight was tirelessly collected and analyzed by these folks. Okay, let me get this little video kind of out of the way. My name is Stephen Whitaker, as Sam has mentioned, and I work as a marine ecologist for the National Park Service at Channel Islands National Park. And this is typically what I look like in the field. Personally, I don't really see the resemblance here, but you can see that other critters tend to find that you need to hunker down in the intertidal if you're going to be there. I spent the past 12 years studying the coastal ecosystems at the Channel Islands, but my passion for marine biology dates way back to when I was a kid. So I was fortunate enough to always live on or near a lake or a coast. And my brother and I even had our own boat when we were out. We weren't even out of grade school. We called it the River Rats. After that, I studied marine biology at the College of Charleston in South Carolina during the late 1990s. And then I received my master's in marine biology from Cal State Fullerton in 2009. And more recently, as Sam mentioned, I returned to school to pursue a PhD at UCSB, studying the causes of decline for important algal populations known as rockweeds. Now, this is me the past week. I wish I could see all your faces right now. I would be remiss if I did not come clean up front about the gobs of time that I spent preparing for this presentation. Now, I've learned that apparently sounding like a scientist is not remotely as cool as I thought it was. So it is my intent tonight to stick to the basics and keep the message simple. So simple is good, but I also think that the best messages are told as stories. So hopefully this one will be just informative and accurate enough to leave you more and not less. So to help with this, I'm borrowing a tip from Randy Olson, an Ivy League marine biologist turned Hollywood film director. And Dr. Olson swears by the ABT principle, and, but, therefore. And the way that it works is something like this. So I'll begin with an explanation of the Rocky Intertidal Zone, what it is, where to find it, how best to explore it. And I will discuss how we monitor the Rocky Intertidal Zone. But the Rocky Intertidal ecosystem is under stress. And I will explain why this is and how we know it. Therefore, why is it important than ever to continue monitoring the Rocky Intertidal ecosystem? And importantly, what can we all do to conserve and protect this valuable sliver of the planet? So by the way, the picture here on the right is of a species of that rockweed that I work on, and the one that I referred to earlier. I like to think of rockweed as the equivalent of kelp for the Rocky Intertidal Zone. It's an incredibly valuable part of the ecosystem, but it has declined throughout a lot of Southern California. So the intertidal zone is between the terrestrial or the land and marine or ocean environments. And it is the area of the shore that is covered by water at high tide and uncovered at low tide. Pictured here, the intertidal zone is the area indicated by the large yellow arrow, and it is below the upper beach and above the surf zone. So you can get the perspective of just how narrow the intertidal zone is in this image taken from the air above Santa Rosa Island. The arrows point to the approximate upper and lower boundaries of the intertidal zone. Intertidal zones can be soft, such as the sandy beach in the top photograph that was taken on the south side of Santa Rosa Island. Incidentally, those sausage-looking objects there in the photo, those are mostly elephant seals and a few California sea lions mixed in. Of course, intertidal zones can also be hardened or rocky, as seen in the lower image of Betcher's Bay on Santa Rosa Island. And there are unlimited combinations of sediment types that make up intertidal zones, from mud and cobble to boulders and bedrock. So many people refer to the Rocky Intertidal Zone as the tide pools. However, some of us intertidal biologist types tend to get a little bit ruffled when these terms are used interchangeably. So tide pools are specific areas within the intertidal zone that retain water when the tide is low. So no two tide pools are identical, but they all provide a glimpse of what life may be like further out to sea in areas generally less accessible to us. In this photo, you are gaining the unique perspective of being half immersed among brightly colored green anemones within a rocky intertidal tide pool at Olympic National Park. So I love these two images of the intertidal zone at Cabrillo National Monument. So I like them because they clearly contrast the same location at high tide, as seen in the top photo, and low tide in the lower photo. And the red arrow is placed on the same landmark in both of the photos for visual reference. The bottom photo shows you what you can potentially see if you arrive during a good low tide. The Rocky Intertidal Zone can often be divided into different zones. This is referred to as zonation. The zones are largely determined by the amount of time that they are exposed to water or air. And they typically host high abundances of key species, such as barnacles, mussels, or various types of algae. In this picture of a Rocky Intertidal site at Santa Cruz Island, you can clearly see different zones. So the spray zone is at the highest part of the shore. And I'm gonna just assume, because I didn't check, but I assume that you can see my cursor move around. Okay, cool. And so this is where species are most accustomed to being dry a lot of the time. Now, below the spray zone is the high tide zone. And this is the area that is only wetted during high tide. And it's composed primarily of barnacles that are very faintly visible in this. Let me get my cursor moving. They're very faintly visible in this picture. It's a white band that occurs here. And, but it's very narrow. In the mid-tide zone is equal parts wet and dry during the tidal cycle, and it generally hosts more species than the zones above. But here in this picture, the mid-tide zone is mostly composed of brown rockweed. It's this band right here. And then finally, the low tide zone is where the greatest biodiversity typically occurs. This area is usually underwater, where it's at least wet most of the time. And in this picture, the low tide zone is dominated by some green surf grass near the water line here, and then some red turf algae. Now, the intertidal zone is highly variable, and it can often be a hostile environment. The marine organisms living in this zone have evolved various adaptations to cope with stress resulting from being out of the water for extended periods of time. Actually, many species are found only within this narrow band of coastal habitat. And some of the stressors for intertidal organisms include intense sunlight and heat, wind, waves, and especially in the lower zones, competition for space and predation. Extensive studies in the intertidal zone have indicated that abiotic or non-living factors associated with stress during low tide, such as desiccation or drying out, determines the species' upper limits, while the biotic or the living interactions, such as competition with other species, sets their lower limits. So that's where they're where they're located in the intertidal zone. All right, so that's all great, you're thinking. The intertidal zone it's a pretty cool place, but I'm sure a lot of you just want to know more about why I impersonate sea stars. And I'm not here to disclose personal secrets, but I will explain the who, what, and the why of intertidal ecosystem monitoring. So the National Park Service has a long history of monitoring ecosystems, both on land and sea. So for instance, at Channel Islands National Park, we've been monitoring the status of natural systems, such as the Rocky Intertidal Zone, since the early 1980s. And more recently, in the early 2000s, the National Park Service established an inventory and monitoring division. And that is, and I'm gonna quote here, it's like a physician for our parks. The I&M division tracks the health of key vital signs, such as water, plants, and wildlife. This information is used to take care of our national parks for this and future generations. So therefore, long-term, well-designed monitoring programs are essential for distinguishing between natural and human-induced changes. And I'll touch on that a little bit more at the end of the presentation. But our national parks are just a really small piece of the puzzle. Other programs run by academic institutions and other government agencies are also monitoring the Rocky Intertidal. Together, we combine efforts to monitor over 200 sites from Alaska in the north to Baja California in the south, using identical methods. Collectively, we are called Marine, the multi-agency Rocky Intertidal network, as I mentioned in the beginning. At the core of the Marine program is a simple and inexpensive, yet statistically sound approach to collecting long-term monitoring data. Now, one of the core protocols we use is monitoring fixed plots. Fixed plots are areas on the shore that we monitor year after year. And here you can see me scoring a fixed plot that is used to track the population trends of target species, such as barnacles or mussels. Each rectangular plot is marked with stainless steel bolts in the corners to help us locate them. And fixed plots were chosen instead of randomly located plots to minimize monitoring effort. The device that I'm holding in my hands is a multi-counter that I use to keep track of all the times that I encounter a species in the plot. Now here you're getting a biologist perspective of what a plot looks like. This is what I'm looking down at. And this white rectangular device we place over the top of the fixed plot is called a quadrat. Quadrat includes a grid of 100 equally spaced points. And we identify every species that occurs under each of those 100 points. So for example, in this plot that I'm displaying, there's a mix of California mussels and let's see, coralline algae, other types of algae. We have some green, what we call sea lettuce or ulva. And so what I would do is I would start in one corner and I would work my way all the way to this corner, and I would identify everything that falls under a point. So in this case, where my cursor is, there's a point, and that would be a California mussel. If I came to this point, I would call that bare rock, and so forth. At each site, permanent fixed plots were established to track the trends for a limited number of species, including barnacles, turf algae, such as the one here I'm pointing to, it's called Endocladia, rockweeds, and mussels are just some example of target species. These organisms provide a range of sensitivity to trampling and other human disturbances. They are all important to the ecosystem, and they are easy to identify and monitor. The larger photo provides a reference for what a typical monitoring site looks like. The yellow text and the rectangles indicate the locations of fixed plots spread throughout the reef. And we sample and photograph every plot once per year. Now, we also continuously monitor temperature at most study sites. And this is accomplished with these small, affordable temperature sensing instruments like this one pictured here. Temperature sensors are typically housed within stainless steel cages or PVC containers like the one that the arrow is pointing to. That protect them against the extreme forces resulting from waves and projectiles such as rocks and logs. The housings are glued to the surface of the intertidal zone in a location that affords some protection and allows us to access them during a moderately low tide. And during each annual site visit, we download or replace the temperature loggers. All right, so enough background. This is really the point in the storybook narrative where the plot starts to thicken. So in other words, well, what have we learned from all of our monitoring and experimentation? So in this section, I will summarize some of the most current knowledge of stressors on the Rocky Intertidal Zone and how this ecosystem is responding to one particular stressful event known as a marine heat wave. Sea temperature strongly influences what species live where. So the average sea surface temperature along the North American West Coast follows a gradient from cool to warm, moving north to south. In this figure, the cooler temperatures are represented by the cooler colors or the blues, and the warmer temperatures are indicated by shades of red. So this north-south gradient in sea surface temperature is the result of a powerful current called the California current that runs north to south along the coast. Now, in the Southern California Bight, where we are located below Point Conception, so I'll run my cursor in this area, there is another current, which is aptly named the Southern California countercurrent. And this current runs in the opposite direction from south to north. And it bathes this region with warmer water compared to areas further offshore where the California current is dominant. Beginning in 2014, a marine heat wave named the Blob formed in Northeast Pacific. So the anomalously warm pool of water shown here as red and purple, it remained in place for 711 days, and it gradually dissipated in 2016 and 2017. Marine heat waves have been documented since 1925, but our sea surface temperature record only dates back to the early 1900s. I mean, that's pretty long, but it's very likely that marine heat waves actually occurred long before the first record. Now, marine heat waves are considered not so great for the critters in the ocean because they typically result in nutrient-poor water that can have profound negative effects on marine life. Unfortunately, marine heat waves are not the only threat to our oceans. So things like invasive species, global climate change, marine debris, overfishing, poor water quality, and altered habitats, they're each significant problems capable of causing extreme damage to our oceans. Climate change in particular is a profound stressor that is altering ocean conditions at unprecedented rates. Increases have been documented for for the temperature and the height of the ocean, as well as the power of waves generated from intense storms. Predictions favor further increases in these conditions, as well as the frequency and intensity of El Nino events and marine heat waves. At the same time, the water is becoming more acidic, oxygen is decreasing, and changes are occurring to the currents. Now, aware that ocean conditions are changing at unprecedented rates, Dr. Pete Ramundi, a marine ecology professor at UC Santa Cruz, analyzed data from more than 200 Rocky Intertidal communities or sites along the North American West Coast. And he looked at the species composition of the communities, and he found that over the course of the 15 years of the data, the compositions of intertidal communities in the north were shifting to look more like the communities in the south, as indicated by this yellow arrow that's on the diagram. These colored dots here on this map, well, they're supposed to represent the locations of the intertidal communities used in the study. And each color represents a unique assemblage of intertidal species. Now, this poleward or northerly change in the intertidal communities is expected due to warming from climate change. What was perhaps most interesting, though, was that not all the communities shifted in composition. Some remained the same over time and appeared to be more resilient or stable. Now, this concept that some intertidal communities were more resilient than others was intriguing, and it actually provided some hope. So for instance, if we could figure out what factors contributed to stability or resilience of rocky intertidal communities, then maybe we could manage for those conditions and improve the status of less stable communities, or at least reduce the rate of change. So that was the focus of this journal article that a group of us recently published in the Journal of Animal Ecology to examine patterns of community stability and identify potential drivers of community change. Now, this effort was led by Melissa Miner, Dr. Pete Ramundi at UC Santa Cruz, along with several National Park Service ecologists and researchers from Cal Poly Pomona and Cal State Fullerton. The article was published last month, and it's open access for anyone who's interested in reading it. For the study, we used data from those fixed plots that were collected annually from 2006 to 2017 at 74 Rocky Intertidal sites spanning from the Olympic Peninsula in Washington State to San Diego, California. The sites were grouped into 4 different regions. The North region, shown here, included 19 sites located in Washington, Oregon, and Northern California. The Central region was comprised of 20 sites from San Francisco to Point Conception. The South region included 16 sites that were located from Point Conception to San Diego. And the last group comprised the Channel Islands region, which had 19 sites located on the northern Channel Islands off the coast of Santa Barbara, California. Now, we also included ocean temperature data from temperature loggers at the sites. All right, so I lied. I added a few graphs in here, not too many. When we plotted the ocean temperature averaged across sites in each region, this is what it looked like. And you're seeing the North region on the left and the Central region on the right. Here, temperature is in degrees Celsius on the vertical axis, and the years of the study are here on the horizontal axis. You can see a little bit more variability in the temperature, in the sea surface temperature, experienced at the Channel Islands here on the left, and the South region over here on the right. And when you overlay the time periods when El Niños and the marine heat wave occurred, and those are indicated by these pink bars, you can see that there were noticeable increases in temperature during these events, especially for the combined effect of the strong El Nino and the marine heat wave during the 2014 and 2015 time period. So that's this thicker bar here. So the next step of the analysis involved determining the extent to which these intertidal communities at each of the 74 sites changed from 2006 to 2017. And so for the purposes of this presentation, I'm just going to keep it simple. I'm going to refer you to our paper to learn more about the statistical procedures that we used. All the details of the statistics are behind the results, and behind the results that I'm going to be presenting now. They can be found in the methods section of the paper. So after determining how much the intertidal communities changed at each site, we split those sites into two different categories. One group included the sites that either did not change or they changed just a very little bit, and we colored those sites blue. The other group included the sites that changed substantially over the course of the survey period, and those sites were colored red. And so you're seeing the map of what those sites look like. The communities that change the most are identified in this map as red symbols, and the communities that appeared more stable over time are shown as blue symbols. And really the take-home message here is that even though there were both red and blue sites in all four of the regions, most of the red sites occurred in the two southern regions, including the islands and the mainland sites below Point Conception. And this implies that the intertidal communities at these sites within the islands and the south regions were less stable relative to the intertidal communities in the central and the northern regions. So I understand when I say that the intertidal communities changed, I realize this is kind of hard to visualize. I mean, what does it look like? So what I've done here is to help with that, I'm showing just one of the hundreds of fixed plots that changed in species composition over time. And this particular plot is located on Middle Anacapa Island. So you're seeing 9 photos of the same plot over time. And it's showing the same plot in 2006 and then 2007 all the way to 2014. Now, keep in mind, this is just one example. And this is one of the more extreme examples of plots that changed in community composition, but I wanted to show it to you to kind of drive the point home. So during the first 3 years of this plot, in 2006 through 2008, you can see the presence of rockweed. It's this golden seaweed here. And then in 2009, all the way through 2014, and I didn't include the pictures through 2017 because I didn't have space, but I can assure you that they didn't look much different than what you see in 2014. The plot is now composed primarily of just bare rock and the pink volcano barnacle. We call it tetraclida. So as I mentioned, we found that the intertidal communities changed the most in the southern regions on both the mainland and the islands. The periods of community change aligned with the abnormally warm periods of sea temperature that occurred during the El Niños and the marine heat wave. All right, so get ready. This is what it looks like in graphical format. Now, I've been told by every person that reviewed this presentation to avoid showing this graph, but just bear with me. I think it may appear scary, but I want to direct your focus to just a couple key points. So let me walk you through it. All you need to know is that each of these 4 colored lines represent the intertidal communities at the 4 regions that we studied. So in this case, blue is for the northern region, green is for central, yellow or orange is for the islands, and red is for the southern region. And if you follow the lines from left to right, you'll see that they all go down slightly. They all do the same, they all do that, but some go down more than others. In fact, the orange and the red lines drop more than the blue and the green lines. And what this means is that the communities of species in the intertidal sites let me start that over. What that means is that the communities of species at the intertidal sites in the islands and south regions changed more from 2007 to 2017 than the communities in the north and the central regions. Furthermore, when you overlay the time periods when the El Niños and the marine heat wave occurred, you can see that the lines dip during these periods. This is especially true for the orange and the red lines, the islands and the south regions. And this implies that the intertidal communities in the islands and the south regions changed more during periods of anomalously warm water than the north and the central regions. Now, unexpectedly, we found that the greatest amount of change in the intertidal communities occurred during the cooler periods of the year for ocean temperature. So this was a little bit counterintuitive, since we expected that the communities were changing during the period of the year when the ocean temperature is the warmest during the summer and the fall. However, it's not the case. They were changing the most during the warm winters. And finally, when we analyzed the data to see if we could predict which intertidal communities would change the most over time, we discovered that mussels and biodiversity played really significant roles. The plots that had high cover of mussels typically remained the same, or they changed little over time, compared to the plots that had less mussel cover. Similarly, the plots that had high biodiversity typically remained the same or changed little over time compared to the plots that had reduced biodiversity. All right, congratulations if you're still here. That was without a doubt a lot of information that I threw at you. So I'm checking the time here. If you give me just a few more moments, I'll sum it all up in this last section. All right, we found that rocky intertidal communities change the most during warm water events, such as El Niños and heat waves. So this is concerning. And it's concerning because global climate change is expected to cause El Niños and marine heat waves to become more intense and frequent. This in turn may cause intertidal communities to continue changing in composition, such that they become dominated by bare rock and warm water tolerant species such as the pink volcano barnacle and red turf algae. And the cover of California mussels may also continue to decline. We found that the intertidal communities change the most in the southern regions on both the mainland and the islands. So unlike storm events where damage tends to be really patchy within and among sites, warm water events can affect broad stretches of coastline. Marine heat waves can occur anywhere in the ocean. So the pattern seen in Southern California during 2014 to 2016 may be seen elsewhere in the future. In the regions that experience the greatest level of community change, the South and the Channel Islands, the cover of mussels appeared to be the most important predictor of community stability. The decrease in California mussels is particularly concerning because they are an important foundational species that provide food and habitat for other species. They're like the glue that helps to hold intertidal communities together. All right, so tell me what it all means. Well, we learned that the rocky intertidal communities are the connection between terrestrial or land and marine ecosystems. As such, the intertidal zone is essential to the proper functioning of both terrestrial and marine ecosystems, because the intertidal zone serves the roles of both food and habitat for a wealth of species. Humans also benefit from a healthy rocky intertidal ecosystem because it provides a highly accessible glimpse of marine ecosystems that are otherwise challenging to explore. Although rocky shores are comparatively minor habitat on Earth in terms of area, they have played a disproportionately large role in our understanding of ecological systems. And additionally, the tide pools provide excellent recreational opportunities and generate revenue from tourism. Oops. So relative to other less accessible marine ecosystems, the intertidal zone is most familiar to and highly valued by society. However, nearshore marine ecosystems face significant challenges at global and regional scales, with threats from both land and ocean. Monitoring composition and abundance of species and understanding their roles in the nearshore ecosystem is essential when responding to and managing present and future threats. I'm obviously reading that. All right. The accessibility of rocky shores provides people with an important connection to the marine environment, but this also makes them vulnerable to injury from human activities. Many factors impact the resilience of ecosystems and their ability to remain stable during periods of stress. Our ability to control many of those factors, such as unusually warm temperature from the water and wave disturbance, is really limited. However, there are plenty of actions we can take to lessen the pressure on the rocky intertidal ecosystem. So for instance, simply reducing our impacts caused by trampling and collecting and flipping over rocks while we're tide pooling, it can make a huge difference, since our intertidal communities here in Southern California receive literally extreme levels of visitation. We also can properly dispose of harmful contaminants that may otherwise make their way into the storm drains and ultimately down to the intertidal zone. And last but not least, please lend a hand, pick up some of the trash. I mean, unfortunately, there's just no shortage of marine debris. And, you know, now that we're easing out of this pandemic, hopefully group beach cleanups are becoming a thing again. So I recommend reaching out to your local park or surf rider chapter to find out when and where those beach cleanups are occurring. I'd like to end by encouraging you all to volunteer for your local national park, if you're willing and able. Our national parks literally depend on the assistance provided by volunteers. From resource protection and interpretation to maintenance and law enforcement, all of our divisions rely heavily on volunteers. And you can find more information about volunteering at nps.gov. All right, acknowledgements. Dozens, if not hundreds, of smiling faces have played an integral role in our long-term intertidal monitoring program. I can't begin to thank everybody, but you all know who you are. And I hope that you know that you are highly appreciated for all your contributions. And I think that's all I have.
All right, and the crowd goes wild, Steve.
Yeah, I'm sure.
Thank you so much for that. It looks like we've got some great questions queued up in the Q&A, so let me pull that up here. And the leader is Interdeep Gill, who has asked a bunch of really, really amazing questions. So we're going to start with his first one, or her first one. How does the type of sediment in an intertidal zone, Bandy or Rocky, affect the biodiversity found there?
Excellent question. There are so many details that I didn't have time to get into that relate to how biodiversity in general changes based on the sediment type. And so, for example, virtually all of our monitoring sites were chosen on bedrock surfaces. So these areas of really wide, expansive, rocky reefs, these big platforms, that's where you're going to see, generally going to see some of the highest biodiversity. And that is in part because you don't have the disturbance of the sediments mixing into where these species live. So when you, for example, if you go to a habitat, an intertidal habitat that is composed of boulders and cobble, you're not going to see nearly as many types of species as you would on a bedrock reef. And that's because those boulders and those cobbles are moving around constantly in the waves. And so the species that are trying to live there just can't stay for long before a big wave comes and knocks them off. And so that's an excellent question. And, you know, on the flip side of that, if you think about a sandy beach, which is still an intertidal zone, as I mentioned, you're going to have high biodiversity, but those organisms are all living within the sediment. So they're called infaunal. They're actually inside the sediments as opposed to on top of the reef, like you would find typically on a rocky intertidal platform. So hopefully that answers your question.
Mm-hmm. So are rising sea levels significant enough to affect the zonation of California's rocky intertidal zones?
Sam, I'm sorry. Can you repeat that? You broke up a tiny bit.
Okay, happy to do so. Are rising sea levels significant enough to affect the zonation of California's rocky intertidal zones?
Another good question. Absolutely. We're not necessarily measuring that yet. We do have plots designed to measure just that. So what we've done recently in the last 5 or 6 years, as I recall, is to put plots, these fixed plots, way up onto the shoreline where we don't see anything living up there yet. And the idea is that if we monitor those over time, we may see things start to move up into those fixed plots. And so it's going to be a very gradual process, but we do fully expect that the species are going to move up the shore as sea level increases.
Are you building a field of dreams for them, Steve?
Yeah, absolutely. And we want to watch them show up.
Exactly. Actually, more like because it's climate change influencing this, a field of nightmares, I guess. All right. Caitlin would like to know, what is the keystone species for your observation? And I know you kind of answered that. It seems like there's several species that you were talking about, like of alga and mussels, but perhaps you could clarify a little bit or even add, are there other keystone species that you guys monitor for as well?
Excellent. Yeah, so I only covered a very small number of species that we monitor over time. And you know, the quintessential keystone species that we monitor is the sea star, the ochre star. And the way that we monitor ochre stars is we set up fixed plots for them as well, but they're big plots. They're not like those little rectangular plots. They're big areas of the shoreline that we inspect every time we go to the monitoring site, and we look for those sea stars, the ochre stars, and we measure them. We measure the longest arm on them, and of course count them. And so we do that. We also monitor for black abalone. We're seeing in recent years that black abalone that, you know, were on the endangered species list, and they still are. Fortunately, we're starting to see some recovery of black abalone populations at the islands. We're not seeing it everywhere, but we are seeing it at a few of the islands. So that's hopeful. Other species that you know, that maybe not as sexy, I guess, are some of the different types of algae that we monitor. And we typically choose species as target species, as I mentioned, because they generally create zones within the intertidal gradient. So rockweed, for example, that makes up most of the mid-tide zone, and so it's very common. It's a common species, and so that's a large reason why we target that species. Trying to think of others. Owl limpets, that's another really cool organism. You know, it starts its life out as a male and then becomes female as it gets bigger. I mean, that's just awesome. And so we have fixed plots to monitor them, and we also measure them as well. So there's a handful of species that we not only count and, you know, see how abundant they are, but we also gather information on their size so that we can see how that's changing over time as well.
Yeah, great, Steve. Thank you. Yeah, I know there's so many interesting species that you study. You can't necessarily go into depth about all of them. That was a great sampling. Would you say the black abalone, we're seeing some improvement there due to robust restoration efforts?
Well, you know, in the case of black abalone, you know, we're trying to work on ways to restore black abalone, but the problem with black abalone that we're encountering right now is that we're not able to spawn them in captivity. So other species of abalone like white abalone and red abalone, no problem. You can have them in an aquarium, and we have very distinct protocols how to cause them to, you know, make them spawn so that we can make more red abalone and black and white abalone. But for one reason or another, black abalone it's a mystery as to what it takes to get them to spawn. And so restoration efforts at this point are hampered based on that. But you know, there are a lot of smart people working on that problem, and we're hopeful. We're getting all the other pieces of the puzzle in place so that when we do finally figure out how to spawn them in captivity, that we'll then know, okay, where are we going to put them? How do we put them out there? You know, and you know, all the details that are going to need to be figured out.
Right. Excellent. Are the El Niños something that have happened throughout history?
Good question. Yes, El Niños are natural. They're that, and the odd thing is, and I actually had to, go back over the literature to make sure that I'm correct in stating that climate change has an effect on El Niños, because El Niños are natural. They've been happening, they've always been happening. So yes, that is true. But as I mentioned, with climate change, they are projected to become more frequent.
Has Marine monitored differences between remote intertidal sites, such as the islands, compared to more urban areas like La Jolla in San Diego?
The beginning of your question, has marine monitoring
Differences between remote intertidal sites, such as the islands, compared to more urban areas like La Jolla?
Yeah. Okay, so two parts of that. As far as monitoring goes, and I think that's what the question was, they're quite different. And I can say that for sure. When I went to Cal State Fullerton, I helped out with Rocky Intertidal monitoring in my lab on areas on the Orange County coastline that are very accessible. We went to the monitoring sites with a group of 10, 15 people. When you go out to remote sites like the islands, we just don't have the capacity to bring more than 2 or 3 people. We have limitations on housing and boating and everything that it takes to get to those sites. So yeah, there are some major differences there in monitoring in terms of what we're capable of doing. Now, and I also want to maybe kind of pivot a bit, because maybe also the part of the question was, are there differences between remote sites and these more populated sites? And the answer there is also yes. And that is because sites that are remote on the islands, they just don't receive the visitation. It's good and bad. The people, people aren't able to get to these sites very easily. But as a result, these rocky intertidal communities don't have the trampling, the picking up of, organisms, turning over rocks. That kind of stuff just doesn't really happen at most of the island sites that I monitor. And so you can really see distinct differences when you go to a site that does occur all the time in the Orange County, San Diego sites, where you generally see really high cover of what we call filamentous algae. So it's the little turf stuff, the little red turf algae that you find all throughout mainly the mid and the lower intertidal zone. That stuff you will see at the islands, but it's much bigger. So it when it's trampled, it actually gets trampled down. And so those are types of things. It's often, when you're at a remote site, you'll see really unique things like nudibranchs. Oftentimes, you don't see those at the more heavily visited sites. So those are the kind of a few of the differences between the visited sites versus the ones that were quite as visited.
For sure, human impact definitely makes a difference. You can even see that at different sites in San Diego compared to Cabrillo, which is protected, versus sites that are not protected, and looking at that, species diversity and all of that. So this is a good segue into Laguna Ocean Foundation's question, which is, given the high visitation rate, as you mentioned, in Southern California, what's the best way to reduce trampling? Is trampling defined as simply walking on the rocks? Maybe you can elaborate and elucidate a little bit there, Steve.
Yeah, it's tough. The Rocky Intertidal Zone, as I explained, it's such a compact area. There isn't a whole lot of space to begin with. I guess my recommendations is, first of all, if you can stay out of the tide pools, that's the number one thing. If you could just try to stay out of tide pools. When you're walking around throughout the intertidal zone, just be really cognizant that, you know, you're walking on animals. It kills me. I have to do this, obviously, to monitor these sites. And, you know, I inevitably am walking on mussels, and sometimes the mussels crack underneath my foot, and it drives me nuts. I hate it, but I have to be there to do this. And you know, hopefully you keep that level of disturbance at a minimum. So, you know, I guess that would just be my only recommendation, is just to try to walk lightly as possible, as if you can. Try, you know, to limit the number of times you pick things up, and just, you know, kind of like keep the same etiquette as you would if you were on a coral reef, for example. You wouldn't want to touch the reef. You just would like to try to admire it from a distance. So.
Yeah, it's a challenge because you want people to enjoy these spaces, but also they do have an impact, right? So, yeah.
Yeah. Yeah, it's just, you know, and to be honest, I have been actually really surprised when you go out to Laguna Beach, or, you know, the San Diego sites, LA beaches, and see how many people are walking around and on these rocky reefs. It's actually really surprising to me and cool that you can go out there and still see, you know, mussels, and still see limpets and barnacles and algae, and, you know, so these things are super robust, but they do get injured, no doubt about it. Trampling has an impact, so it's just good to keep that in mind.
Tread lightly. Ken would like to know, how can managers use the results from monitoring to be better stewards?
Excellent question. I knew Ken would come up with a great question. Well, so, you know, one of the things that we hope to be able to show is that if you can show that the Rocky Intertidal community is already stressed, you know, like marine heat waves, for example, is a major stressor that we're finding for these intertidal organisms. You know, what we would like to do is to try in our best way to limit other stressors, whatever stressors that we have some control of. And, you know, so some of those might be trampling, you know, and if we can show that, you know, certain parts of the coast are really sensitive to these things, then, you know, maybe that we could limit some of that disturbance that we're in control of. And that's kind of, I think, the best recommendation that I could have in terms of taking a management action, would be to, you know, maybe there's certain times of the year where if we were to just limit some of the visitation to a site, then it would have a chance to kind of, you know, it rebound a little bit. So I think that would probably be my recommendation.
Right. Putting some guidelines there and some limitations to let the populations kind of bounce back a bit, give them some time to restore. Nick would like to know, what are some important reasons that might motivate people to learn and do more for our rocky intertidal ecosystems?
Well, I mean, the place is so cool to begin with. I mean, I don't know if I even have to advertise it much, but yeah, so, you know, I think it's important to, first of all, to visit the place. Like, you know, we're talking about all of the, you know, reducing visitation, but at the same time, it's a very important habitat for people to visit, because this is really where almost everyone I know is introduced to the ocean. You know, it's to get out to the kelp forest, you know, you're going to have to put your bathing suit on or a wetsuit and, you know, maybe a scuba tank. Whereas you can do that in the Rocky Intertidal Zone. You can get that experience of seeing a real marine ecosystem right there in person. So, you know, I mean, if that in itself, it should be the motivation for wanting to preserve these really special ecosystems.
Larry would like a little clarifying point here. Is it that more stable areas allow for more mussels to grow, or is it that mussels create the more stable environment?
So that's a very good question that I kind of been grappling with as I've been putting this presentation together. And it's the best way that I can explain it is that in the southern region, where there were mussels present at sites, those sites remained what we call stable, or they remained pretty much the same throughout the study period, which in our case was 2007 to 2017. Whereas the plots or the sites that started out with low cover of mussels, those changed a lot over the course of the study period. So that's kind of like the best way I could think about it.
Like more research is needed too. What are the greatest challenges to long-term monitoring of intertidal areas?
Oh, well, so challenges, well, I would have to say that the challenge is we're trying to figure out what's to come. So we're trying to make sure that our protocols are documenting these things that might happen in the future. At the same time, we want to continue doing the protocols that we've always done. So the challenge is doing as much as you possibly can with very limited resources, and not only resources but time. Keep in mind that we can only monitor these places when the tide is out, and I didn't include some slides in this presentation. I ended up taking them out that explain, at what time of year that we go out, because low tides in this region, and I'm speaking Southern California, are really only good, and I say good meaning that you can get out and actually work from October through May of the next year. And then during the summertime, like right now, all the low tides are going to happen during nighttime. And, some intertidal researchers go out at nighttime. Fortunately, it's not safe enough for me to go out in the islands at nighttime, but you would have to do that. And so in summary, it's doing as much as you possibly can with limited resources.
Right. Often the challenges of science, period, right?
Yeah, and life in general.
So we are running a little low on time. Do you have time for just like one or two more questions?
Absolutely.
Okay, great. And I'm sorry for those of you who I didn't get to your questions tonight. You all had some really excellent questions today. So let's talk about a little bit about sea stars. Someone would like to know how you can account for sea star withering.
Yeah, unfortunately, in 2014, there was what we call a sea star wasting disease event that occurred. And this was coast-wide. I mean, it was an unprecedented disease, marine disease outbreak that occurred from Washington State, or even slightly above, all the way down through Mexico. And this widespread event affected multiple species of sea stars, up to 8 or 9 different species of sea stars. And in some cases, actually had a really significant impact on the population. So in the case of sunflower sea stars, we just recently got an observation of one that might have been seen, or probably was seen, at Santa Barbara Island. But other than that, we haven't seen a sunflower star since 2014. It also had a big impact on the ochre stars that are in the intertidal zone. That's the main one that we document or we monitor. And the way that we monitor for withering or wasting disease is by way of these fixed plots that I explained. But we have added, we've kind of tweaked the protocol a tiny bit so that we're also, in addition to measuring the sea stars, we're also inspecting them for signs of disease. And in this case, for sea star wasting disease, they become inflated. They have this inflated appearance, and they start to develop sores and lesions on their arms. This whole thing happens in a very rapid amount of time. So within 2 to 3 days, a sea star can go from having one sore on one of its arms to multiple sores on arms to losing their arms completely and then dying. It's a terrible event that occurred, but we since then we continue to monitor sea stars and to inspect them for signs of the disease happening again. Just to be clear, sea star wasting disease, that was an unprecedented event that happened in 2014 and 2015, and yet those types of events have happened in the past, but just not to the spatial, to the extent that it happened in 2014. They did on much smaller scale. We saw waste disease in sea stars in the past.
Are you seeing any recovery for sea star species?
Now, as I mentioned with the sunflower star, we're not seeing recovery. There may be some recovery occurring really far north, like in Washington and above. I'm not as, I haven't heard recently what the observations are up there. But I know in our region and slightly in Central California, Northern California, they're not seeing recovery of sunflower stars. Now, ochre stars, we are seeing recovery in certain places, but it's very patchy. It's not happening everywhere. Above Point Conception in Central California, they're seeing at times, they'll see lots of baby ochre stars. But we didn't see that down at the islands and the Southern California mainland. We would see a few babies, but nothing like that was seen in further north. At this case, I would like to, when I explain this, it's good to have some context. In the past, at the island sites, we would see upwards of 100 to 1,000 ochre stars at one intertidal site. Oh. Whereas these days, at the most, I've seen 20 or 24, somewhere in that ballpark for a site. Typically, our intertidal sites don't have more than a handful of ochre stars at this point. But that doesn't mean that they're not out there. It just means that we're not seeing them at our monitoring sites. The times that I do go and get some time to go get underwater and look around, I have seen places where you'll see dozens of ochre stars in the same, in a very relatively small area. So they're out there. They're just not in the numbers that they used to be.
Hopefully that'll change over time and they'll come back. So let's end tonight's wonderful talk with a kind of like a positive message here. Steve, I'd like to know, what's the thing you love most about being a marine ecologist? What really floats your boat?
Wow. Well, I can't even explain like how cool it feels to be out on the remote islands and know at times that I'm one of the few people that's even on the island. And the fact that I'm supposed to be there. I'm being paid to be there, and I'm down in one of the places that people go to explore on their off time. And I also find it just so incredible, such an incredible feeling to think that I'm responsible for documenting what's going on there. And so that alone just really, I guess, in your words, it floats my boat, to think that I feel sort of important. So that's pretty cool.
But remember, with great power comes great responsibility, right, Steve?
Absolutely.
For these species. All right, I want to thank our event sponsor, Cabrillo National Monument Foundation. Thank you so much for supporting this event and paying for the captions so we can make this accessible. Of course, I'd like to thank our wonderful speaker, Stephen Whitaker, for speaking tonight. Thank you so much, Stephen, for spending all this time talking about the current research and your experience in the Rocky Intertidal. And I want to thank all of you for coming to this Naturally Speaking talk. We'll be back in about a month or so, probably shifting focus again to our pollinator palooza. So we'll be back to pollinators and plants. But thank you again, everyone, and have a wonderful night. Goodbye. Take care, everyone.
Bye.